Hello friends, this video on molecular basis of inheritance part 33 is brought to you by examfear.com. No more fear from exam. These parts. So now let us look at the function of the Lac Operon. Now let us start from the beginning. This is the promoter, this is the operator, and this region is the structural gene. So as I said, the regulatory gene will be functional all the time. So the regulatory gene will get expressed continuously. That means the regulatory gene, that is I gene, is always active, so it is always synthesizing repressor. So the repressor which is responsible, the specific repressor for Lac Operon is being synthesized all the time. So the repressors are available all the time. Now what will happen? This repressor will bind to the operator region of Lac Operon. So this is the operator region, the blue region, and the repressor will come and it will fit with the operator region of the Lac Operon. Now as soon as the repressor binds to the operator, what will happen? Now when the RNA polymerase comes and tries to bind with the promoter, because that is what it has to do. It has to bind with the promoter to start its job of transcription. Now when the RNA polymerase tries to bind to the promoter, it is prevented by this repressor. That's because due to its presence, the promoter is made inaccessible to the RNA polymerase. So as a result, the RNA polymerase is not able to continue with its job of transcription. So now what happens? The process of transcription stops, that is, it is inhibited, and as a result, these genes are turned off. The genes Lac Z, Lac A, Lac Y, they are all turned off. So all those enzymes which are responsible for fermentation of lactose, they will not be produced. Now, do you think that we needed all those enzymes all the time? No, right? We need those enzymes only when lactose need to be fermented. But right now there is no lactose inside the cell. So we really do not need the enzymes to ferment the lactose. So it is perfectly fine even if these genes are turned off right now. So that means this serves the purpose of controlling the expression of these genes. So right now all these genes are turned off. Now as long as the repressor will remain binded to this operator region, these genes will remain turned off. As soon as the repressor comes out of the operator region, then the RNA polymerase will be able to perform its job, and then those enzymes will start getting synthesized. Clear? Okay. So now let us see what happens. Now as we know, every organism needs energy. Now every organism will have different sources of energy. So the preferred source of energy, if you talk about in case of E. coli, will obviously be a monosaccharide like glucose, where there is no complication of breaking it down further because it is the simplest form of a carbohydrate. So obviously glucose would be the preferred source, but in case glucose is not available, in that case also it will need energy. So how do we provide that energy? So in that case, lactose can be provided to the growth medium of E. coli. Now when lactose is provided to the growth medium of E. coli, what do we want? We want lactose to enter inside the cell of E. coli. So how will lactose enter? So for this situation, the enzyme permease, which we were talking about, right? So that permease enzyme increases the permeability of the cell for entry of lactose, and that is how transport of lactose into the cell take place. Now you might say that when these genes are turned off, that means none of these enzymes are getting synthesized, whether it is permease or it is transacetylase or it is galactosidase. So from where is this permease coming in? Now, very little amount of these enzymes remain there, because it is not necessary that when the genes are turned off, it is completely turned off. Sometimes they are like turned off very minutely. So very small amount of these enzymes might be produced or might be present inside the cell. So with that small amount of permease, the permeability of the cell can be increased enough to fetch in more lactose inside the cell. Now, what happens? So permease will help in transporting lactose into the cell. So now you will start getting lactose inside the cell. Now once lactose start coming into the cell, What is the need? The bacteria or E. coli needs to utilize energy. So for that, lactose need to be fermented. So you will need the enzymes which are responsible for fermentation of lactose. So let's see what happens next. So now what happens? Now lactose starts entering inside the cell. So inside the cell, we have now lactose molecules. So here these purple colored molecules, they represent the lactose molecules. Now lactose initiates the turn on of the genes. The presence of these lactose molecules initiate the turn on for the genes, and that is why in the Lac Operon, lactose is termed as the inducer, because it induces the initiation, or it induces the turning on of the genes. So let us see how does it induces the turning on of genes. So lactose acts as the inducer, so something which induces something. So we'll see that. So what happens? One of these lactose molecule will bind to the repressor. So the repressor which was bound to the operator. So one of the lactose molecule will bind to the repressor, and as soon as the lactose molecule binds to the repressor, there is some structural changes in the repressor, as a result of which the repressor is inactivated. So the repressor comes out, the bonding between the repressor and the operator breaks, and the repressor come out of the operator. Now as soon as the repressor comes out, what happens? The path is all clear for RNA polymerase. So RNA polymerase will quickly bind to the promoter, and then it will continue with the process of transcription. Now as soon as it continues with the process of transcription, what will happen? A strand of mRNA will be synthesized, and this mRNA in turn will help in the synthesis of the proteins, and the proteins are nothing but the enzymes which we spoke about: galactosidase, permease and transacetylase. So all these enzymes will then be produced, right? So now what happens? The genes are turned on. So what exactly initiated the turning on of the genes? The lactose molecule, and that is why lactose acts as inducer. Now what is going to happen? As soon as you start getting these enzymes will start performing their job. Right? Now, once the synthesis of enzymes had happened, these enzymes are produced, they will start doing their job. And what is their job? The job of galactosidase is to ferment lactose. That means it will start taking lactose and break it down into glucose and galactose. So that means galactosidase will actually start reducing the number of lactose which is present inside the cell. So gradually what will happen? Again, the lactose molecules will start disappearing, because beta galactosidase will hydrolyze lactose into glucose and galactose. Now once the lactose molecules are all gone, again this repressor will bind to the operator, and again as the repressor binds to the operator, what will happen? The function of RNA polymerase will again be blocked, and therefore the transcription will stop. So therefore every time lactose is present, whenever lactose is present, the genes will be turned on. So under what scenario genes are on? Genes are on when lactose is present, and that is why lactose is an inducer and not inhibitor, right? And also when the repressor is attached to operator. Repressor is not attached to operator, right? This is the scenario when the genes will be turned on. And under what scenario the genes will be off? When lactose is absent. And whenever lactose is absent, what will happen? The repressor will be attached to the operator. So this is how the genes are being controlled. This is how the expression of genes, whether these genes will get expressed or not, whether the synthesis of protein will take place or not, that is all dependent on the repressor and also on the inducer. So this is how gene expression is regulated. So this was just one example of Lac Operon which takes place in E. coli. There are other operons also. For example, there is a Trip operon which acts where tryptophan plays an important role. Similarly, there is another operon called the Val operon. So there are several operons which are functioning in the E. coli. But here we have just discussed one operon just to give you an idea about how it takes place. So looking at its function, you can now see that this is an absolute example of negative regulation. So whenever the repressor attaches to the operator, it has a negative impact. That is, the process of transcription is stopped. That is, the genes are turned off. So that is why the repressor is an repressor, and it is going to cause. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.